DNA, or also known as the deoxyribonucleic acid, which is present to all living organisms and is referred as their blueprint. Do you know that DNA testing is used not only to identify a person's identity, but also to authenticate food, such as caviar and fine wine?
DNA was first observed by a German biochemist named Friedrich Miescher in 1869, but for many years, researchers did not realize the importance of this molecule. It was not until 1953 that James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin figured out the structure of DNA, a double helix which they realized could carry biological information.
Watson, Crick, and Wilkins were awarded the Nobel Prize in Medicine in 1962 for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. Franklin was not included in the award, although her work was integral to the research.
Similar to the way the order of letters in the alphabet can be used to form a word, the order of nitrogen bases in a DNA sequence forms genes, which in the language of the cell tell cells how to make proteins. Another type of nucleic acid, ribonucleic acid, or RNA, translates genetic information from DNA into proteins. Nucleotides are attached together to form two long strands that spiral to create a structure called a double helix. If you think of the double helix structure as a ladder, the phosphate and sugar molecules would be the sides, while the bases would be the rungs. The bases on one strand pair with the bases on another strand. Adenine pairs with thymine, and guanine pairs with cytosine.
DNA molecules are long, so long, in fact, that they can fit into cells without the right packaging. To fit inside cells, DNA is called tightly to form structures we call chromosomes. Each chromosome contains a single DNA molecule. Humans have 20 pairs of chromosomes, which are found inside the cell's nucleus. A person's DNA contains information about their heritage and can sometimes reveal whether they are at risk for certain diseases. DNA tests or genetic tests are used for a variety of reasons, including to diagnose genetic disorders, to determine whether a person is a carrier of a genetic mutation that it could pass on to their children, and to examine whether a person is at risk for a genetic disease. For instance, mutations in the BRCA1 and BRCA2 genes are known to increase the risk of breast and ovarian cancer. An analysis of these genes in a genetic test can reveal whether a person has mutations.
The importance of DNA. It has been argued that the discovery of DNA as well as our understanding of its structure and functioning may well be the most important discovery of the last century. The effect of the discovery of DNA on scientific and medical progress has been enormous. Whether it involves the identification of our genes that trigger major diseases or the creation and manufacture of drugs to treat these devastating diseases. In fact, the identification of these genes and their subsequent analysis in terms of therapeutic treatment has ultimately influenced science and will continue to do so in the future.